Great. Thank you very much. Hi, everyone. My name's Paul Rennert. I'm president and CSO of Aleta Biotherapeutics. It's a cell therapy company located outside of Boston in Natick, Massachusetts. And I'm not going to talk about Aleta today. Instead, I'm going to tell you what to look for at ASCO coming up in a few weeks. I put this together last night. The abstracts actually published at 5 p.m. East Coast time last night, and there was just sort of a flurry of activity around some of the high-profile talks. However, this is not going to be inclusive. This is really going to reflect mainly my interests, as you'll see, and we'll just go through it together over the next 15 or 18 minutes or so. So as I started to frame the talks up that I wanted to share with you, I broke them into two major categories. One is the cell therapies. As I mentioned, Aleta is in the CAR T cell space. We also work with a class of biologics called CAR T engager molecules, and I wanted to find time to talk about T cell engagers and NK cell engagers and bispecifics of various sorts. But I realized that was going to just be too much time for what we have today. So instead, I focused in on a few of the novel immune checkpoints. And I'm glad I did. I mean, this is a field, which has really been stalled for 6 or 8 years or so. And we've been waiting for good news on new immune checkpoints that we can prosecute. And we got some last night out of the ASCO abstract. So I'll talk through a couple of those with you. And then there's just 2 or 3 things that I wanted to share kind of in a more rapid fire format. So a lightning round, so to speak. And we'll get through as many of those as we can as time allows. So let's just go into the cell therapies. And I want to set a little bit of a context, right? Because it's been a really newsy month or two in cell therapy. What we see across the biotech and pharma landscape are lots of new technologies. This was certainly prominent at ASGCT, the American Society of Gene and Cell Therapy meeting, which was last week or the week before. And we're seeing, of course, an endless drumbeat of new company launches, many of those companies being built around the new technologies that are being developed. So that's the good news, right? It's a very exciting space, and it's continuing to grow rapidly, and it's attracting a lot of investment from both from private equity and from large pharma. On the other hand, we have seen some stumbles. So Precision Bio is a company that's been around for a number of years, and about, 3 weeks ago or so, they went through an upheaval in management and moving assets that had been partnered back into the portfolio, and just creating a little bit of a buzz around what was going on at Precision. And I'll share some of their data with you in a few minutes. It's fair to say Selectus, which has really been a pioneer in the allo CAR T cell space that we're going to talk about, they had quite a rough showing at ASGCT around one of their programs. So that cast a little bit of shadow over the allo space, which I'll do my best to remove in the next few slides. And just yesterday, we got this abrupt resignation of really extremely talented executive running Iovance, and she resigned yesterday on the heels of a regulatory delay that the company was experiencing. So, a lot of news, both good and bad, as we go through the news cycles. But, at the end of the day, it's clinical data, not the news cycles that tell the tale for each of the therapeutics that are being developed. And I want to share a couple of those stories with you. So Allogene came out with their data on Allo 501A. This is an allogeneic CAR to CD19 being trialed in diffuse large B-cell lymphoma and follicular lymphoma. I suspect that's transformed follicular, but I'm not sure. You'll note here that there's a reference to ALLO-647. That's their proprietary anti-CD52. So that is a lymphodepletion antibody built along the lines of Campath, for those who remember that molecule. It's fair to say that anti-CD2 mediated lymphodepletion is really aggressive, and that's one of the issues that at least I have with allogeneic approaches to date. The good news is that Allogene showed some really nice response data, and at least in the 3 to 6 month time frame, the responses they saw were quite good and comparable to some of the early data we saw in the autologous space. Now, the question will be what happens after this 6 month time point where they've got, a good collection of CRs. Will they continue to see relapses, or will we start to see the formation of a long tail, right, as we did in the Yescarta, lymphoma trial, the Zuma-1 trial? Now, the worry around this technology is that it really does require deep lymphodepletion. So chemotherapeutic lymphodepletion on top of the anti-CD52. So these patients lose their immune system. But the data are promising, and we'll see more of it as this trial reads out. I mentioned Precision. They presented data on PBCAR 191. This is their allo anti-CD19 CAR, slightly different patient population, diffuse large B and mantle cell. So, the good news for Precision, they did report some CRs in the preliminary data. I think the bad news was the tolerability, which was clearly a little bit mixed. Whether this is due to the deep lymphodepletion that's needed, or other things, I'm not sure. But out of 13 patients treated, they lost one patient during the treatment regimen. So, we didn't see a lot in the abstract itself. I think we need to stay tuned to see what data they present actually at the meeting in a few weeks. One of the things I noticed last night, they released a press release. They're already looking past this particular therapeutic to the next one in their arsenal, which has more modifications to the cell to make it more allo. And this will be gene editing modifications to their next therapeutic. William Blair put out a table of responses across the allo spaces. We've seen it to date, and they're quite similar from CRISPR to Allogene to Precision. The rubber will meet the road here as the durability readout is extended beyond, these early data, and as we understand tolerability and the need for redosing. I think redosing, although it sounds simple, I doubt very much that it is, and that'll be another complexity as we evaluate the space going forward. So one more cell therapy snippet. Arcellx had presented some teaser data at ASGCT. That ASGCT data is shown below. They're going to show more data at ASCO, and I think the results to date look good. This is an anti-BCMA CAR T cell for multiple myeloma. They're getting a lot of majority of patients are responding, and many of the responses appear to be durable. So this looks like a winning combination for Arcellx, at least in this early data set. So let's talk a little bit about novel immune checkpoints. I think the big news, as you may be aware, is Bristol's data on the combination of anti-LAG3 relatlimab with nivolumab in frontline metastatic melanoma, showing really a nice improvement in progression-free survival. This is important because the data are as good as Ipi-Nivo, and that's been the benchmark, right, in melanoma. And as you can see in the table from Jacob Plyth's write-up, the tolerability data is much, much better than Ipi-Nivo when you compare the two combinations. And this seems to be, really a winning drug, at least in melanoma. This casts, a little bit of a shadow on everybody else that's trying to make, IO combinations with immune checkpoint inhibitors in melanoma, but it's always good to move the bar higher. That means we're doing better for patients. So good news all around on this study from Bristol. There was another LAG-3 abstract from Regeneron, and there they looked at melanoma as well in combination with their anti-PD-1. And, it seemed to me, just reading the abstract, that there seemed to be more adverse events and less efficacy from this drug than the Bristol drug. And it's not clear to me why. They're both IgG4s. They're both paired with anti-PD-1s. They're both in melanoma. And yet this one does not appear to be clearly better than their PD-1 alone. And I think this begs the question as to whether it's inferior to the Bristol drug or not. More data will tell the tale, as always. Moving on, Innovant also had a LAG-3 inhibitor. This is in solid tumors now, advanced solid tumors. Really, modest responses here for this LAG-3 antibody. One confirmed PR in ovarian and some stable disease. Some stable disease is in the dose escalation. Not much better in the phase 1b part of the trial. Again, significant adverse events, a little bit different from what Bristol showed. So I think what we're going to need to see out of ASCO is an across the trial comparison of these, as well as the other LAG-3 inhibitors that are out there, including Merck's. Immunotep has LAG-3 inhibitors, and of course others. So a field that's rapidly expanding, and I think will digest the data as it comes. The other big news last week was that Bristol did a big deal with Agenus for a TIGIT, a set of TIGIT assets. If you haven't followed TIGIT, you can go. I wrote about it recently. You can see the link here. There's a lot of molecules. They have somewhat different properties. So we've got wild type G1s with effector function, mutated G1s without effector function, G4s. Seattle Genetics or Seagen has a enhanced ADCC TIGIT. So people are coming at this in a bunch of different ways. There's not a lot of data to share from the ASCO abstracts. The one that's there is from BeiGene. This is a small study in advanced solid tumors in combination with their anti-PD-1, and really not much happening here. So the excitement generated by Bristol, in doing the Agenus deal is not reflected in this early data from BeiGene. Again, it's early days. The BeiGene mAb is an intact IgG1. We'll have to wait and see if that turns out to be a good choice or a bad choice. This field is evolving. So let's move in the next couple of minutes to just some additional abstracts. So this is one that caught my attention. This is atezolizumab. This is the anti-PD-L1, really moving into the adjuvant setting post-chemo, really in stages of non-small cell lung cancer that we don't often see isolated in these kinds of trials, showing a really nice improvement in hazard ratio in the patients who were PD-L1 positive. This, speaks to one of the differences between nivolumab, for example, which typically does not get selected on PD-L1 expression, and the Merck drug, which does, and Atezo, which does. Sometimes it's difficult to compare across trials because patients may or may not have been selected by the PD-L1 biomarker, but this looks like compelling data in non-small cell lung cancer, adding to what we already see in that space in different stages, and here in the neoadjuvant setting, which is really nice, or the adjuvant setting, which is really nice. Okay, so here's a drug class that I find really, really interesting, and a drug that is maybe less interesting. So the drug class are the cytokines. There are a bunch of these being developed. Many are being developed as fusion proteins with antibodies. Others are being pegylated, and so on and so forth. And here's a molecule from Alkermes. This is designed not to bind to Tregs, but only to bind to CD8 positive T cells, effector cells, CD4 effector cells, and NK cells. And it's suffice to say that the data here are a little bit underwhelming. And I think for me, what this says is there's more work to do in the cytokine space. We'll see a lot over the next few years. So that's the exciting part. So, I'd like to see more data out of Alkermes at the meeting. But right now, at first blush, this is probably not competitive against that LAG-3 program, for example. This being, of course, in advanced solid tumors, not just in melanoma. So maybe an unfair comparison. Another one, this is another winner. This is the Mirus drug, really a clever bispecific that has a strong effector function, binds to HER2 and HER3 well, I'm not sure it's simultaneously, but binds to both HER2 and HER3. What that does is prevent HER2 from escaping from therapy by creating HER2, HER3 heterodimers. That's a classic way that HER2 positive solid tumors can escape from therapy. There's EGFR upregulation, there's HER2, HER3 heterodimerization on the cell surface to escape therapy. This is a really nice program targeting a specific set of patients having NRG1 fusion proteins that induce this heterodimerization. Really excellent response data in pancreatic cancer, where it's hard to see anything at all. The data are summarized here in a table, again from William Blair. Now I'm just about out of time, and I'm just about out of lightning round summaries. I did want to mention that the speaker who's coming up next, Dr. D'Souza from Ultimovacs, would likely declare himself a lightning round winner based on their data in a phase 1/2 study of a melanoma of their therapeutic with pembrolizumab. And that's the end of my time and the end of my slides. So I'll stop there and hand it back over to the control room. Thank you very much.